Neurological Disorders Lesson 4.5, How do Circuits Regulate Their Output? The goal of this lesson is to demonstrate how different arrangements of excitatory and inhibitory connections can regulate the output of a neural circuit. At the end of this lesson, students should be able to describe how circuits use excitation and inhibition to control output and distinguish between the process of feed-forward inhibition and feedback inhibition. We'll achieve these goals through a Socratic discussion as well as a student model of feedback and feed-forward inhibition. To prepare for this lesson, you'll need to review the key scientific concepts presented in it. They include regulatory mechanisms used to maintain homeostasis, including feedback inhibition and feed-forward inhibition, as well as the biological clock, its components, primarily the suprachiasmatic nucleus, its connections to the preoptic nucleus, the pineal gland, and the sleep-wake circuit, and its functions. We'll also discuss the biological clock disorders, including jet lag, seasonal affective disorder or SAD, and Monday morning blues. You can review this scientific content in the background reading provided to you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in the lesson. The teacher manual or lesson plan provides a minute-by-minute explanation of lesson structure, including instructions on how to manage the discussion and activity. And the student workbook provides additional explanation for your students. You'll need to be sure to print the lesson worksheet, activity instruction cards, and activity signaling cards for your students. You may also opt to use colored yarn or string for the student model. The key points of the lessons due now is the biological clock, its components, primarily the suprachiasmatic nucleus or SCN, and its connections to the preoptic nucleus, the pineal gland, and the sleep-wake circuit, as well as its functions. We'll get to this key point by reviewing the homework. In the Do Now, students review last night's homework with a partner, being sure that they can describe how the suprachiasmatic nucleus, the SCN, connects to the flip-flop switch or sleep-wake circuit, as well as describe how the activity of the SCN is regulated. The key points of the discussion are, again, the biological clock, its components, connections, and functions, as well as regulatory mechanisms used to maintain homeostasis, including feedback inhibition and feed-forward inhibition. We'll get to these key points with a Socratic discussion. The discussion begins with a review of last night's homework, specifically how the SCN connects to the sleep circuit, both when there is light and when there is darkness. You'll then discuss how the activity of the SCN is controlled by introducing the concept of feedback inhibition. The activity of the SCN is ultimately controlled by the expression of clock genes, which encode for clock proteins, which then regulate behaviors and activities. These clock proteins then inhibit the expression of clock genes, creating a feedback loop. This slide is animated, so you can step through each stage of the process with your students. The key points of the activity is again the regulatory mechanisms used to maintain homeostasis, including feedback inhibition and feed-forward inhibition. We'll get to these key points with a student model. The activity begins by asking students why do traffic lights have both green and red lights. This question is framed to help students prepare for the models they will be creating. The models use red and green as symbols for inhibitory and excitatory synapses. But unlike traffic lights that have both green and red lights, we'll be talking about synapses, which will either be inhibitory, like red lights to slow down activity, or excitatory, like green lights to speed up activity. The activity then continues with two student models to demonstrate how neural circuits use a combination of excitatory and inhibitory connections to control their output. Now caution, these student models require both the appropriate setup and time to allow student observers to watch the effect of the actions within the model circuit. The first student model, outlined on slide number 9, uses 4 students to model feedback inhibition. Each student within the model is given the appropriate signaling cards and could also be given string to help show and demonstrate the connections it's making with other neurons within the circuit. The second student model, outlined on slide number 10, uses 2 additional students to include a section of feed-forward inhibition. Again, the students are all given the appropriate signaling cards and may benefit from also having string to demonstrate where they are signaling to. After acting out the models, slide number 11 includes animations to allow you to step through the actions of the model in a slide format, something that some classes have found very useful. The key points of the wrap-up are that feedback and feed-forward inhibition are critical for regulating the output of neural circuits, as well as to address biological clock disorders, including jet lag, seasonal affective disorder, and the Monday morning blues. We'll get to these key points with a Socratic discussion. In the wrap-up, we'll refocus on the fact that feedback and feed-forward inhibition are important, especially because our neurons do not have a single output and a single input as we were modeling today. 99% of our neurons look like the neurons pictured on this slide with many inputs. The wrap-up also addresses why we need feedback and feed-forward control at a behavioral level, using examples of biological clock disorders that occur from dysregulation of our sleep-wake circuit, including jet lag, seasonal affective disorder, and Monday morning blues. The key point of the homework is that feedback and feed-forward inhibition are critical for regulating the output of neural circuits. We'll get to this key point with a worksheet. For homework, students will complete part 2 of their lesson worksheet, which is designed to get them thinking about controlling excitation, which will lead into our discussion of epilepsy in tomorrow's lesson. If students need additional help with this homework, they can refer to the workbook reading associated with lesson 4.5. Students are often challenged by the content in this lesson, so it is important that you understand the concepts well so that you can guide your students. You can find more in the teacher primer, teacher manual, and student workbook. One of the most common questions teachers get is where else might I have seen feedback and feed-forward inhibition? An example commonly used to describe feedback inhibition is the thermostat, because when temperature gets too cold, the thermostat turns on your heater, which produces heat until it reaches a set point, and then the thermostat turns the heater off again. Since the product, heat, turns off its own production, this is an example of feedback inhibition. If we stay with the theme of heating and cooling, an example of feed-forward inhibition would be a forecast for cold weather, which would prompt you to proactively close your windows, turn off your AC, and maybe turn on your heater. Or likewise, a forecast for hot weather prompts you to proactively open your windows, turn off your heater, and turn on your AC. At the end of this lesson, students should appreciate that the activity of our neural circuits are balanced through a variety of regulatory mechanisms, including feedback inhibition and feed-forward inhibition. The concept of inhibitory control sets the stage for the next lesson that examines the clinical consequences of abnormal inhibition, and for later units in which we'll examine more complex circuits. Don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members and we'd be more than happy to help you.